Okay, so let's talk about how you're going to run your surface area to volume IA. So I'm going to run through kind of the steps of what the experiment looks like, and then I'm going to run through what you are going to be turning into me. Okay, so what you're going to be getting is agar, which is going to be pink. It has been stained with phenolphthalein, which is a pH indicator. When you add a base to it, sorry, not a base, an acid to it, it turns clear. And we saw this in class. So basically, you're going to use a cork borer, I don't know how to spell it, borer, I think this is it. And you're going to cut out tubes of this agar. And the cork borers come in a variety of sizes. You need to have 3 sizes of cork borers. Okay? These 3 sizes are going to be your independent variable. Alright, and the cork borers are labeled on the size, on the sides of the actual borer with the size. That is not a measurement. You need to measure the diameter of the cork borers. That will be your independent variable. Okay, so you're going to have your pieces of agar. They're going to be different sizes. I'm not drawing these very well, but you get the idea. Okay, and you want them to all be the same length. That would be the easiest thing to do. Okay, I made this look even bigger, but let's see. Alright, so same length. You need to record that because you're going to need to figure out the surface area and the volume of these 3 tubes. And you're going to do 3 trials. So, which means 3 repeats. So you're going to have a total of 9 tubes. That's what you need total. Okay, and then these guys are pink, so let's make them pink. We're going to make them red because I have that easily available. Oh, it's pinkish. Okay, so these are going to go into one concentration of acid. Okay? Okay, so you're going to take a beaker, plop them in, and then you need to time them. You're timing until they are no longer pink in the center. Okay. Okay, the acid is going to go in to the tubes via diffusion. Okay? Because remember, it's not water moving, it's particles of the acid moving. Okay. Okay, so there's some measurements you need to take in order to be able to calculate what you need to calculate. So ultimately, you are calculating the surface area of each tube, and you're calculating the volume of each tube. Okay? These are calculated values. But make sure you get the data, like the measured data that you need in order to calculate these values. And remember, you need to actually measure the diameter of the tubes. Don't just rely on the value on the cork borer, because that is not a measurement. Okay. So keep in mind that you need what you need in order to measure these 2 things. Okay, and just to remind you, I'm just going to erase this really quickly. The units for these, for surface area, are going to be centimeters squared, and for volume it will be centimeters cubed. Okay, so beyond calculating the surface area and the volume, you're also going to calculate 2 other things. You're going to calculate the surface area to volume ratio, which is literally dividing surface area by the volume. Okay, and then the other thing you're going to calculate is something called rate of diffusion. Now we've talked about diffusion, and this is what we're doing right now, right? So for this, you're going to have the volume divided by time. Okay? because it's always a distance divided by time, so that's really what this is here. When you're doing your time, you need to make sure it's either, your whole time is either in minutes or seconds. Not like minute dot seconds. You need to convert it so that it's one or the other. Otherwise, Excel or Google Sheets will not know how to deal with this. Okay, so let's talk about what you're actually turning into me. Number one, you're going to have a title. Now your title should include your IV and DV. And probably something about the fact that we're doing this on agar blocks. You want to give some context in your title. Often a title is going to look like a research question. Okay. Then you're going to include 2 tables, at least. So you need to have table 1. You need something with raw data. Incidentally, raw data is not a title for a table. Tables should always have titles that include the independent and dependent variable. So raw data is not a title that's acceptable. So I expect to see a table with a raw data, which in this case is anything that you have not processed. And then I also expect to see a calculation section. So calculation should include formulas that you've used, any formulas with processing that you've used to process, and examples. One example for each one is good. Don't forget units in your examples. Okay, then I expect to see a second table that has your processing. So probably, again, this is not a title, you might even have more than one table. So probably one that has surface area to volume ratio worked out. And you're not doing this to the average, you're doing this for each sample and then finding the average. Find the average at the end. Okay, so you're going to have that. And then you're also going to have your rate of diffusion. Now when we're talking about actual surface area and volume, that can go up here with raw data. Even though it's processed, I would be fine with that. And volume. Okay, if you want to have it with the processed data, it technically is, either way would be okay with me. Okay, then I expect to see 2 graphs. Okay, the first graph is going to be surface area to volume ratio, and it's going to be time. Okay, just to be clear, the surface area to volume ratio is after you have divided them. And then I expect to see some scatter plot, line of best fit. So that's the first one. The second graph I expect to see would be surface area to volume ratio, and then rate of diffusion. Okay, same idea. Boop. Don't forget your units, your labels on your axes, etc. Okay. Okay, and then the other part I expect, so this all was data, and then I expect a conclusion. So you're going to summarize your results first. And you can use data to do this. Now this is not an analysis section. This is where you're sort of answering a research question should you have one. So in this case, it's how does surface area impact time, and how does surface area impact rate of diffusion. Use your data, pick out the most important points to put in there, and then you're going to explain, this is very key, you're explaining your results. So you're going to talk about how does what you know about surface area to volume ratio relate to what you found? Why does what you found look like what it is? You may well need to go and do some research on surface area to volume ratio and how that works. I recommend the textbook or BioNinja. So use knowledge about. Okay. And that's what I'm really expecting for this lab. There are many other components in a lab, but this is your first one, so we're going to keep it relatively short and sweet. Okay, that's it. Thank you.